Hello and welcome to day 2 of our Bioinformatics Internship program. Today we will dive into the fascinating world of bioinformatics, starting with an introduction to its core principles. Today we have an exciting agenda ahead. Let's dive into the topics that we will cover today. Firstly, we will begin with an introduction to molecular biology. We will discuss the key concepts and significance of this particular field, exploring its fundamental techniques and applications that are essential for understanding the molecular basis of life. Next, we will move on to the introduction to bioinformatics. Here, you will learn about the definition and scope of bioinformatics and its critical importance in modern science, particularly in analyzing and interpreting the biological data. Following that, we will explore the link between the molecular biology and bioinformatics. We will examine how these two disciplines complement and also enhance each other, providing integrated applications in the field of research. This section will illustrate the synergy between the molecular biology and the bioinformatics field in advancing the scientific knowledge. We will then discuss the roles and skills of a molecular biologist versus a bioinformatician. This discussion will differentiate between the roles and skill sets of these particular professionals, highlighting their unique contribution to scientific research and how their expertise overlaps and diverges. Looking towards the future, we will speculate on the future of bioinformatics. We will consider a potential advancement and emerging trends for the next decade, discussing on how these developments might shape the field and impact science and technology. Next, we will dwell into the career prospects in bioinformatics. We will provide insights into the job opportunities, industry demand, educational pathways, and professional growth within this dynamic field, helping you better understand the various careers prospects available over here. We will then provide an overview of the biological databases, which are very much important resources for the bioinformatics research. You will learn about different types of databases, their applications, and also understanding their role in managing and accessing the biological data. A specific focus will be on the NCBI database today. We will explore the features and functionalities of this important resource, showing how it serves as an essential tool for accessing a wealth of biological data. We will also demonstrate the retrieval of nucleotide sequence. You will learn how to effectively retrieve a sequence from this particular database, an essential skill for a bioinformatics analysis. Finally, we will guide you through the process of saving a FASTA sequence. This practical step is crucial for managing and analyzing the genetic data, providing you with the hands-on experience in bioinformatics techniques. By the end of today's session, you will have a solid understanding of these key areas. So let's dive in and explore these fascinating topics together. Molecular biology. So molecular biology is a branch of science that majorly focuses on understanding the structure and function of the molecules that constitutes to a living organisms. So molecular biology examines these interactions between DNA, RNA, and proteins. So DNA contains the genetic blueprint for life. RNA translates this particular information into proteins, and the proteins perform a wide range of essential functions within the cell. So molecular biology employs various techniques such as PCR, maybe for amplifying of DNA, gel electrophoresis for separating the obtained DNA fragments, and also CRISPR-Cas9 for gene editing. These tools have revolutionized our understanding of cellular processes and have numerous applications in the field. In medicine, molecular biology has led to breakthroughs in diagnosing and treating genetic disorders, cancers, and infectious diseases. In agriculture, it is used to develop genetically modified organisms, which we today call as GMOs, with enhanced traits. Overall, molecular biology provides crucial insights into the molecular mechanisms of life, which is driving advancements across multiple scientific fields. So coming to bioinformatics, bioinformatics integrates biology with computer science and information technology to analyze and interpret the biological data. It utilizes the computational tools and algorithms to maintain larger data sets, including the genomic sequences and protein structures. By applying these tools, researchers can uncover patterns and relationships within the biological data that inform fields such as genome sequencing, phylogenetic analysis, drug discovery, and also personalized medicine. Advancements in this particular field and data science continually propels bioinformatics forward, expanding its application in the healthcare, agriculture, and also the environmental sciences. This interdisciplinary field plays an important role in understanding the complexities of living systems and also addresses the global challenges through innovative computational approaches. So, to decipher this particular term called as bioinformatics, it is derived from the combination of biology, information, mathematics, statistics, and computer sciences. It is a multidisciplinary field at the intersection of life science and computational technologies. Each component of its names reflects an important aspect of the methodology and scope. Coming to biology, at its core, bioinformatics deals with biological data, that is, the genetic sequences, protein structures, and also the biological pathways. These data form the foundation of understanding the molecular basis of life and biological processes. Information. Information theory and the data management are very much important in bioinformatics for storing, organizing, and retrieving vast amount of biological data very efficiently. This aspect ensures that the data can be analyzed and interpreted very effectively. Coming to mathematics. So the mathematic principles are used in bioinformatics to develop algorithms and models for analyzing the biological data. So techniques such as sequence alignment, phylogenetic analysis, and statistical modeling rely heavily on the mathematical concepts here. Coming to statistics, statistics plays a very much important role in bioinformatics by providing methods for analyzing the biological data sets to extract meaningful insights and draw valid conclusions. It helps in identifying patterns, correlations, and significant differences within the biological data. Next, last coming to the computer sciences. So, finally, computer science provides the computational tools and techniques necessary to process and analyze this large-scale biological data. So, this includes software development, database management, and also the algorithm design to support various bioinformatic applications. So, together, these components enable bioinformatics to advance our understanding of biological systems, accelerate biomedical research, and facilitate innovations in healthcare, agriculture, and also the environmental sciences. So, bioinformatics continues to evolve, driven by ongoing advancements in the technology and majorly interdisciplinary collaboration across these fields. So, molecular biology and bioinformatics synergize to advance biological research by integrating the experimental data with that of the computational analysis. To put in simple terms, molecular biology generates the data on DNA, RNA, and proteins, while bioinformatics provides tools to manage, analyze, and also interpret this data very efficiently. So together, they uncover the genetic variations, predict protein structures, and finally elucidate the complex biological pathways. This particular collaboration enhances our own understanding of molecular mechanisms, facilitating drug discovery, enabling the personalized medicine, and also driving innovations across various scientific fields. A molecular biologist is like a detective in the world of biology. They use traditional lab techniques to investigate the fundamental components of life, that is, the DNA, RNA, and proteins. They conduct experiments, analyze the genetic sequences, and study how these genes work to better understand the biological process at a molecular level. So molecular biologist work is mostly hands-on. It involves tasks like isolating genes, manipulating the DNA, and observing how molecules interact within the cells. On the other hand, bioinformatician's role is more like a digital scientist. They specialize in using computers, mathematical algorithms to study biological data. So bioinformaticians analyze massive data sets which contains of genomic sequences, protein structures, and other biological informations. By applying computational methods, they identify patterns, predict how the gene actually functions, and also uncover the insights that can lead to new discoveries in medicines and biotechnology. Together, molecular biologist and a bioinformatician form a powerful team, where one generates the raw data through experiments, while the other processes and interprets this particular data using sophisticated computational tools. Their collaboration is essential for advancing scientific knowledge, from maybe understanding to genetic diseases to develop a personalized treatment based on an individual genetics makeup. So, in the next 10 years, bioinformatics will be expected to see significant advancements across various fronts. In personalized medicine specifically, bioinformatics will play an important role by analyzing individual genetic profiles to tailor medicines or treatments for diseases like cancer. So, researchers will use advanced computational tools to identify these genetic mutations driving the cancer growth. They also will be able to predict patient responses to different therapies and develop targeted treatments that are more effective and also have fewer side effects. So, in cancer research specifically, bioinformatics will continue to integrate genomic data from large scale studies to uncover maybe genetic factors influencing cancer development and also the progression. This data driven approach will enable the researchers to better understand the molecular mechanisms of cancer, identify new biomarkers, and for maybe early detection of this cancer, and discover potential therapeutic targets for novel treatments. Moreover, bioinformatics will contribute to accelerating drug discovery and development of stimulating drug interactions within the biological molecules and predict their efficacy. This computational modeling will streamline the drug screening process, leading to a faster identification of promising candidates for clinical trials. Overall, bioinformatics is set to revolutionize the cancer research and treatment over the next decade. Leveraging both big data and the computational analysis to drive innovations that improve patients' outcome and advance our research understanding in these complex diseases. Navigating the job market with a specialized degree like bioinformatics can feel daunting. You might wonder where this particular degree can take you in the world of scientific careers. Understanding the diverse jobs available in the bioinformatics is an important key. This field not only offers you many exciting and well paying career opportunities, but also majorly puts you at the forefront of scientific breakthroughs and new discoveries. As a bioinformatician with a bachelor's degree in bioinformatics, you will apply your expertise in biology, computer science, and information technology to analyze the biological data. You will tackle complex data sets like genomics, proteomics, using algorithms and computational models to actually predict and interpret the biological patterns. Your work will play an important role in advancing personalized medicine, understanding the genetic diseases, and also developing the innovative therapies. A bioinformatics scientist, his role is well suited for those with a bachelor's degree in bioinformatics. Your work will primarily involve analyzing the biological data to gain insights into the genetic sequences, protein functions, and also other biological systems. So, as a bioinformatics scientist, you will conduct detailed genomic and proteomic data analysis, develop and refine the algorithms for data interpretation, and also collaborate with the researchers and scientists in the field of biology. Your responsibilities include designing and implementing the experiments, interpreting the complex biological information, contributing to the development of a new computational tools and methods, and also publishing research findings in scientific journals. The role of a bioinformatics analyst involves the practical application of bioinformatics tools and techniques to analyze the biological data, supporting research and development in various scientific and medicinal fields. Finally, as a bioinformatics analyst, your duties will revolve around the processing and analyzing of larger data sets, such as genomic sequences or protein structures. Here, you will use bioinformatics softwares and tools to identify the patterns, make biological predictions, and support scientific research. Your analytical skills will help bridge the gap between the raw data and the actionable scientific insights, which plays an important role in advancing biological research and improving the health outcomes. So, coming to databases, a database is an organized collection of structured information or data which is stored electronically, typically which can be accessed and managed using specialized softwares. In the context of bioinformatics and biomedical research, databases play a crucial role in storing, managing, and providing access to vast amounts of biological and biomedical data. These data sets are designed to be searchable and often contain information such as DNA sequences, protein structures, genetic variations, scientific publications, and also clinical data. Databases serve as repositories of knowledge, allowing researchers, scientists, or clinicians to store and retrieve their data efficiently for various purposes. This purpose can include from genomic analysis, drug discovery, disease research, and understanding the biological systems. They can range from simple collections of data to complex systems that integrate the diverse types of information, facilitating the interdisciplinary research and collaboration across the scientific community. Biological databases. These are the organized collections of biological data structured for the efficient storage, retrieval, and analysis. There are three types of biological databases: primary, secondary, and specialized biological databases. Coming to the primary databases, here it is stored in the original format, raw data generated from biological experiments and observations directly. Examples include GenBank for DNA sequences, PDB, that is a protein data bank for protein structures, and SRA for sequencing data, which is a sequence read archive. They serve as a repositories for fundamental biological data such as nucleotide sequences, protein sequences, and raw experimental data. Researchers deposit their data directly into these databases, which are curated and maintained to ensure data integrity and accessibility to the scientific community. Coming to the secondary databases, these are the databases which aggregate, integrate, and also curate the data from the primary databases or any other resources for that matter to provide additional annotations or analysis. For example, UniProt for protein sequences and annotations, Ensemble for genome assemblies and annotations, and RefSeq for curated nucleotide sequences. They enhance data from primary databases by providing value-added information such as functional annotations, gene classifications, and cross-references. So researchers rely on secondary database for comprehensive and curated information that facilitates the detailed analysis and interpretations of biological data. Coming to the final specialized biological databases, it majorly focuses on specific biological domains, organisms, or a type of a data, providing a detailed and curated information tailored to research for particular areas. For example, Flybase is for Drosophila research. RGD, that is a rat genome database for rat genomics, and PharmGKB for pharmacogenomics data. They cater to specialized research communities by offering in-depth data relevant to specific organisms, diseases, and pathways, or experimental techniques. Researchers working within these specific fields or study particularly organisms rely on these specialized databases for comprehensive and specialized data resources that support focused research and analysis. So together, these databases support diverse research endeavors and facilitate advances in the understanding of biological processes, diseases, and genomic variations. Coming to the NCBI database, the National Center for Biotechnology Information operates under the US National Library of Medicine, which is a part of the National Institute of Health, that is the NIH. It is established in the year 1988 and government funded. NCBI hosts a vital database essential for both fundamental and practical life science research. It serves as a critical hub for bioinformatics tools and services. Key databases within these NCBI include GenBank, which stores the DNA sequences, and PubMed, a comprehensive repository of biomedical literature. NCBI database is a crucial resource in the field of bioinformatics and biomedical research. It serves as a centralized repository of biological data encompassing a wide range of information essential for understanding the genetics, genomics, and other related fields. At its core, the NCBI database includes several key components here. Number one is GenBank. This is the primary database within the NCBI, housing annotated nucleotide sequences, that is the DNA or RNA, submitted by the researchers worldwide. So GenBank sequences are extensively used for genetic research, genome assembly, and comparative genomics. Coming to PubMed, it is a comprehensive biological database of biomedical literature. So PubMed here indexes millions of research articles from journals, conferences, and also books. It provides access to abstracts and also full text articles, facilitating literature reviews and research in various medical and biological disciplines. Coming to BLAST, that is a basic local alignment research tool. It is widely used bioinformatics tool today available through the NCBI database. It allows the researchers to compare their own nucleotide sequences or protein sequences against databases to identify similar sequences. BLAST helps in determining sequence homology, evolutionary relationships, and also the functional annotations. So the other databases and tools that NCBI hosts are various specialized databases and tools for sequence analysis, protein structure prediction, and genome browsing, and more. These resources include tools like PrimerBlast for designing the PCR primers, ORF Finder for identifying the open reading frame in nucleotide sequences, and Genome Data Viewer for visualizing genomic data. Data submission and public access. So researchers can directly submit their own sequences and data to the NCBI database, contributing to the collective knowledge base accessible to the global scientific community. So many of the NCBI's resources are freely available, are accessible to the public, promoting transparency, which is very much important for any researcher, and collaborations in the scientific research. Overall, the NCBI database plays an important role in advancing the scientific discovery and understanding the biological processes. It supports researchers worldwide in exploring genetic variations, studying disease mechanisms, developing new therapies, and also addressing the global health challenges through robust data analysis and collaborations. So coming to the NCBI web page, the home page of the NCBI website serves as a gateway to a vast array of biological and biomedical resources. Here is an explanation of what you might find in the home page. Search bar. So prominently featured at the top, allowing users to search across NCBI databases and resources using the keywords, accession numbers, gene names, or any other relevant terms. There is a navigation menu which offers quick links to various NCBI resources such as PubMed, gene, nucleotide, protein, and other specialized databases like structure and genome. Featured tools and services include the highlights popular bioinformatics tools and services offered by the NCBI, such as BLAST, Primer BLAST, and PubMed Central. There is also a section for latest news and updates, which provides updates on the new datasets, tools, and resources added to the NCBI databases, as well as announcements related to research, events, and policy changes in the biomedical field. Educational resources. So it offers links also to tutorials, webinars, documentation to help users navigate and utilize the NCBI database effectively for research and education purposes. Access to databases. So there are direct links or search options for accessing major NCBI databases like GenBank, PubMed, and others, allowing the users to explore data and information related to genetics, genomics, and biomedicine. There is a section for user support, which provides links to help resources, FAQs, and contact information for technical support and assistance with using the NCBI tools and databases. So overall, the NCBI database or the home page is designed in such a way that the central hub for accessing and exploring the biological data, tools, and resources are critical for research, education, and discovery in the field of biotechnology, genetics, and the biomedical sciences. In the home page, locate the drop-down box. So first, look for the search bar or the drop-down box at the top of the home page. It may be labeled as all databases or search. Click on the drop-down arrow. Click on the arrow or the text to open the drop-down menu. This action typically displays a list of different databases or search options that is available on the NCBI database. Now, select the nucleotide option. Scroll through the options in the drop-down menu until you find nucleotide or any other similar term related to nucleotide sequences. Click on this option to select it. Now, perform your search. So after selecting the nucleotide, you can enter your search query. It can be gene name, accession number, or any other keyword related to the nucleotide sequences. Press enter or click on the search button to initiate your search within the NCBI nucleotide database. So by selecting the nucleotide option, you direct the NCBI website to specifically search and retrieve the information related to nucleotide sequences, which includes the DNA and RNA sequences stored in the databases like GenBank. So this helps you focus your search and find relevant biological data for your research and study purposes. Enter the accession number here. Type or copy paste the accession number you want to search for into the search bar. Accession numbers are unique identifiers assigned to biological sequences or sequences records in the NCBI databases, such as GenBank or PubMed. Now, after entering the accession number, click on the search button or press enter on your keyboard. So this action will initiate the search process and retrieve the specific record associated with the accession number you entered. Now, after performing the search, you will be directed to the search results page This page will display a list of records or entries matching your search query. Now, locate the specific record or entry you are interested in reviewing. This could be identified by the accession number you entered. Later, click on the FASTA option. Now, on the record details page, look for options related to the sequence data format that says FASTA. Once you have selected the FASTA format, the sequence data will be displayed in the FASTA format on your screen. That is the ATGC format. You can then copy the sequence data or download it for further analysis. It can be for sequence alignment or comparative genomics. The FASTA file format is a simple format commonly used to store and share the sequence information. When you download sequences from the database such as an NCBI, you usually want FASTA files. So these FASTA files will be saved in .txt extension in your computer system. The first line of a FASTA file starts with a greater than character followed by a name or description of the sequence. Subsequent lines contain the sequence itself, that is the ATGC format. A short FASTA file might contain just something like this. Now, a FASTA file can contain the sequence for a single or an entire genome or even more than one sequence. So, if a FASTA file contains many sequences, then for each sequence it will have a separate header, starting with a greater than character followed by the sequence itself. So here in the picture you can see this is what a FASTA file with a single sequence and multiple sequence looks like. You can store up to 100 or more than 100 sequences in a single FASTA file. Coming to today's assignment, number 1, log into NCBI. Access the NCBI website using your registered email ID. So once you logged in, take a screenshot of the home page that clearly displays your name or email ID to verify your access. Once you register, your name or your email ID will be displayed on the top right corner of the web page. Number 2, retrieve the gene sequences. Using the provided GenBank accession numbers, access the NCBI databases to retrieve the corresponding gene sequences. Save the gene sequences in a separate .txt file. It's important to ensure that txt files are named descriptively, possibly including the gene name or the accession number for better clarity. Number 3, retrieve the FASTA sequences. Now, choose at least 3 genes of your interest from any organism. Use NCBI's tool to retrieve their FASTA sequences in the particular format. Save each FASTA sequence in an individual text file. Again, name these files appropriately to indicate which gene sequence they contain. Now, this assignment aims to familiarize you with using NCBI resources to retrieve genetic data, practicing the data organization, and also to ensure proper documentation of the sequence file for genetic research or the scientific research or educational purposes. That's it for today. Thank you for participating in today's session. Please ensure your assignments are emailed to me on time. If you have any questions about the assignment or anything else, feel free to contact me through WhatsApp. I'm here to assist you whenever you need. Have a wonderful day. Thank you.